Okay, this video is going through the answers to these Hardy-Weinberg AQA past paper questions. Before we actually get on to doing the questions, I need to triple check that you have a few things really clear in your head. I need you to make sure you can tell me what P, Q, P squared, 2PQ and Q squared are all codes for. You need to have these 5 codes crystal clear in your head. Can you tell me aloud, or think the answer in your mind, what is P a code for? P is a code for the frequency of the dominant allele. That dominant allele can exist in 2 genotypes. It can exist in homozygous dominant individuals and it can exist in heterozygous individuals. So it's just how common that allele is within the whole population. Q, tell me what that is. It's the frequency of the recessive allele. And can you tell me whereabouts that recessive allele could be found in a population? It could be found in homozygous recessive individuals or heterozygous individuals. So that's how common this allele is amongst the whole population. These ones, you're getting a bit muddled with at times. P squared is the frequency of homozygous dominant individuals. So how common are the homozygous dominant individuals within that population? 2P squared is the frequency of the heterozygous individuals, and Q squared is the frequency, just missed a line out, haven't I? Frequency of homozygous recessive individuals. I need you to have those codes crystal clear in your head. 5 different frequencies measuring how common either a certain allele is or a certain type of individual is. So learn those off by heart. And then we need to spot them in our questions. So the allele for tongue rolling, capital T, is dominant to the allele for non-tongue rolling. Okay, so this is our P and this is our Q. The dominant allele frequency, the recessive allele frequency, well, the frequencies will come up in a bit. In a population, the frequency of the allele for tongue rolling, T, is 0.4. So what this is saying here, will you tell me which one of those numbers, or letters, sorry, is 0.4 equal to? It's the dominant allele, and it's saying how common, how frequent that allele is, so it's P. So that's literally P equals 0.4, that's what it's saying. Use the Hardy-Weinberg equation to calculate the percentage of people in this population that are heterozygous for tongue rolling. Whenever it says that line, you must first of all write down the equation. Don't take shortcuts because you will, and people I've seen do it, have lost marks. Then we need to figure out, what are we trying to find out? The percentage of people in the population that are heterozygous. So within those 3 groups of people, which one are we trying to find out? This group of people, because they are my heterozygous individuals and I want to know what they are as a percentage. Usually with a Hardy-Weinberg question you start off with a Q and then backtrack to find this one, but we're given P. So how are we going to get to the point where we can find 2PQ? Well, we have a rule that we know that P plus Q is 1. That's just a fact. So if we know that, then we know Q must be 0.6. So 2PQ is 2 times 0.4 times 0.6, which is 0.48. And then it says what percentage of the individuals are that, so 48%. Okay, and then the next one, snails. The shell of the snail may be unbanded or banded. The absence or presence of bands is controlled by a single gene with 2 alleles. The allele for unbanded B is dominant, so B is, we're looking at our P individuals here, and the unbanded ones, little b, so that's Q. Population of snails contain 51% unbanded snails. Use the Hardy-Weinberg equation to calculate the percentage of the population that will be heterozygous. So again, first thing I always do is write my equation down. Then I identify what I'm trying to find out. I'm trying to find out the percentage of population that will be heterozygous. So it's these people I'm trying to find out. What do I know? 51% of these snails are unbanded. Unbanded snails are dominant. So some of you just thought that these ones were unbanded. It's the homozygous dominant and the heterozygous that will look unbanded. And that's 51% of that population. Oops. Which means that this Q squared must be the remaining 49% of the population. I'm trying to find out 2PQ, so I know that Q squared is 49% or 0.49 because we want to do it as a decimal, don't we? Therefore, I can find what Q is, and therefore I can find what P is. Should have brought my calculator down with me. And then I can multiply 2 times P times Q, and I get 0.42. So that's 42% of the individuals. Okay, let's have a look at the Tay Sachs question then. Right, Tay Sachs is a human inherited disorder. Sufferers of the disease often die during childhood. The allele for Tay Sachs is recessive to the allele for unaffected individuals. So the Tay Sachs one is recessive, so that's Q. And the kind of unaffected people, alleles are going to be the P ones. The diagram doesn't exist. In a human population, 1 in 1,000 children have Tay Sachs. Use the Hardy-Weinberg equation to calculate the percentage of the population we'd expect to be heterozygous. It's the same question again, basically, isn't it? So I'm going to start off by putting my equation down. I know 1 in 1,000 children had Tay Sachs. Tay Sachs is recessive. So only these children here have Tay Sachs. They have the disease. So I know Q squared is 1 in 1,000 children. So I know Q squared is 1 in 1,000. Therefore, Q is the square root of 1 in 1,000. I want to find 2PQ, so I need to find what P is. So I know P plus Q equals 1. Therefore, P is 1 take away square root of 1,000 because I don't have my calculator with me. And then I can multiply together 2 times P times Q and get my answer. Now, because that is going to involve a bit of rounding, there's a bit of variation, a bit of a range in the mark scheme of things that you're allowed. So you can have anywhere between 5.8 and 6.2. And then the actual percentage of heterozygotes is likely to be lower in future generations. Explain why. Well, remember, Hardy-Weinberg said this equation will always give you the same frequency of individuals with every generation if 5 conditions are met. Now we know with these children, if you have the disease, you it says at the top of the question somewhere, sufferers often die in childhood. If you die in childhood, that means you're not going to get to reproduce and pass on your genes. One of those conditions for Hardy-Weinberg is that alleles are not selected for or against. The recessive allele here is being selected against because it kills people. So if you have the dominant allele, you're more likely to survive. So the frequency of heterozygous is going to get lower because there are fewer and fewer individuals with those recessive alleles reproducing. Okay, let's look at the cat's question. Polydactyly in cats is an inherited condition in which cats have extra toes. The allele for polydactyly is dominant. in a population of cats, 19% of them had extra toes. Use the Hardy-Weinberg equation to calculate the frequency of the recessive allele. So it's asking me to calculate Q, isn't it, in this population. So first thing I always do is write down that equation. And then what numbers do I have? I know 19% of cats had extra toes. If you've got extra toes, you've got dominant allele. Some of you just thought this is the cats with extra toes. These cats had extra toes because if you're homozygous, dominant or heterozygous, you will have extra toes. So this is 19%, which means that Q squared must be my remaining 81%. So if I know Q squared is 0.81, I can find Q is the square root of 0.81, which is 0.9. Okay. Right, malaria question. Sea otters were close to extinction at the start of the 20th century. Following a ban on hunting seal otters, the size of the population began to increase. Scientists studied the frequencies of two alleles in a population of sea otters. The dominant allele T codes for an enzyme, so that's P, isn't it? The other allele, T, is recessive, that's Q, and doesn't code for an enzyme. In a population of sea otters, the allele frequency for the recessive allele was 0.2. So that is basically saying Q is 0.2. The allele frequency for the recessive allele. Frequency of the recessive allele is Q. That's not talking about the frequency of homozygous recessive individuals, it's the frequency of the allele. It's not Q squared, it's Q is 0.2. Use the Hardy-Weinberg equation to calculate the percentage of homozygous recessive sea otters in the population. So P squared plus 2PQ plus Q squared is 1.0. What am I trying to find out? I'm trying to find out the percentage of homozygous recessive sea otters. That's these ones, isn't it? What percentage are Q squared? Well, I know that Q is 0.2, therefore Q squared is 0.4, and it's asking me to do it as a percentage, isn't it? So 4% of the sea otters are likely to be homozygous recessive. What does Hardy-Weinberg principle predict about the frequency of that allele in another 10 generations? It predicts it's going to stay the same if those 5 conditions are met. It's only a 1 marker, so we don't need to talk about the 5 conditions for this one, but yeah, the frequency should stay the same. Several years later, the scientists repeated their study on this population. They found that the frequency had decreased, so the Hardy-Weinberg principle is not holding up here. A statistical test showed that the difference between the two frequencies was significant at the P equals 5% level, 0.05 level. Use the terms probability and chance to explain what this means. Most of you had the right idea, but you read that as less than or more than it is equal to. So we just need to say the probability that the difference in allele frequency occurred by chance is 5%. Okay, that's all it really wants. Probability and chance, we've done that. The probability that the difference in allele frequency occurred by chance is 5%. Not is less than, not is more than 5%, it just is 5%. Ordinarily, you would be talking, wouldn't you, about things being significantly different or not. It doesn't need it this time. Right, cat's question. What does the Hardy-Weinberg principle predict? Most of us got 3 marks on here. It predicts that the allele frequency will remain constant with every generation, providing 5 conditions are met: no mutations, no selection, large population, isolated population, random mating, no migration. Bizarrely, it only wanted you to have one of those 5 conditions for the third mark, but I think that's a bit cheeky. So I'd definitely make sure I say provided 5 conditions are met, for example, and then quote as many as you can remember, personally, if I had time. Right, we've got some allele frequencies in this table. So these are allele frequencies. These are either Qs or Ps, depending if they're dominant or recessive. We're not looking at individual frequencies. We're not looking at homozygous, heterozygous individuals. We're looking at allele frequencies. We've got 3 different cities, and we've got a couple of different alleles we're looking at. White cats are deaf. Fact. Would the Hardy-Weinberg principle hold true for cats? So basically, are those 5 conditions going to hold true for white cats? Well, the deafness is irrelevant to certain things like mutations, large population, genetic isolation, mating at random, no migration. But because these cats are deaf, they're unlikely to survive. They're going to get run over or something like that. So the Hardy-Weinberg principle doesn't hold true because the white cats are unlikely to survive, therefore they won't pass on their alleles, so you will get a reduced frequency of the white allele with type. What is the evidence from the diagram, from the table, that agouti and blotched are alleles of different genes? Agouti and blotched. Let's find those 2. So how do we know that one is not P and one is not Q? Like, for instance, they must both be Q or both be P. What do we know about P and Q? P and Q always have to add up to 1, don't they? It could be... how am I trying to say this? With a gene, let's say they were the same alleles of the same gene, non-agouti and blotched, there might be another allele involved that's just not in this table, because it does say the frequencies of some alleles. The only evidence where we can prove that they're definitely not the same gene is if they add up to more than 1. So these alleles here in London for these 2 add up to more than 1. These add up to more than 1. This doesn't, so we can't really use that as evidence. But in Paris and London, the allele frequency for non-agouti and blotched add up to more than 1, so they can't be P and Q. They must both be P or both be Q, something like that. Hair length in cats is determined by a single gene with 2 alleles. The allele for long hair is recessive, so that's Q, isn't it? The allele for short hair is dominant, so that's P. Use the information in the table and the Hardy-Weinberg equation to estimate the percentage of cats in London that are heterozygous for hair length. Show your working. So P squared plus 2PQ plus Q squared is 1. I'm trying to find the percentage of cats that are heterozygous. This is coming up a lot, isn't it? Right, so what do I need? I need a P or a Q, don't I, to try and work this out. I've got no information here at all. No numbers in this question. Hair length. London. Let's go back to this table and find hair length and London. London, long hair, 0.3. Okay, long hair. Long hair is recessive, isn't it? And it's 0.3. So Q is 0.3. That's what my table tells me. Let's check it was 0.3 and 0.33. Thought I'd missed something. Okay, so I can tell from the table Q is 0.33 because that's the frequency of the recessive allele in London. Therefore I can calculate P because P plus Q is 1. Therefore I can tell what P must be. And then I can multiply 2 times P times Q, and it wants a percentage, doesn't it? So I need to times by 100. So I end up with 44.22%, but 44% is fine. It will give you, oh no, ignore that, sorry. Right, rats question, I think this is second last one. warfarin is a substance which inhibits blood clotting. Rats which eat warfarin are killed due to internal bleeding. Some rats are resistant to warfarin as they have the WR allele. Can we remember what this notation signifies? When you've got a capital letter and then you've got another capital superscript to it. A bit of revision from chapter 17. It means codominant, doesn't it? Rats have 3 possible genotypes. In addition, rats with WR require very large amounts of vitamin K in their diets. If they don't get that, they will die due to internal bleeding. How can resistance suddenly appear in an isolated population of rats which is never exposed to warfarin before? Well, new genetic changes occur by mutation. So you just want mutation on that line. A population of 240 rats was reared in a lab. They were all fed on a diet containing an adequate amount of vitamin K. In this population, 8 rats had the genotype WSWS, 176 had WRWS, and 56 had WRWR. Use these figures to calculate the actual frequency of WRWR in this population, so you're working. This is not a Hardy-Weinberg question. It would say use the Hardy-Weinberg equation too. This is just about genes. So I've got 3 options. Get them in the right order. WS, there were 8 rats. WS, there were 176 rats. And WR, there were 56 rats. I know that's exactly what it says there, I just find it easier to see it like that. Use the information to calculate the actual frequency of the allele WR in this population. Right, so these guys here have got no WRs in them, have they? These guys here have got 1, and these guys here have got 2. Alright, so there's 176 rats. Each rat has got 2 alleles. So there are 176 alleles that are WR in this population. These guys here, there's 56 of them, and they've got 2 of those alleles each. So that's 112 alleles. These ones got none. So that adds up to 288 alleles in total. A frequency is like a fraction or a decimal, isn't it? So 288 alleles, WR alleles in that population, out of how many total alleles are there in that population? Well, each of these rats have got 2 alleles because they're diploid organisms. So there's 240 rats in total, therefore there are 480 alleles in total. And this is saying 288 out of the 480 alleles are the WR ones, which is 0.6 as a allele frequency. The diet of the rats was changed to include only a small amount of vitamin K. The rats were also given warfarin. How many rats out of the 240 rats are likely to die within a few days? So they're given a little bit of vitamin K, but not a lot, and warfarin. Let's have a look who's going to die. Well, these are resistant to warfarin. Resistant to warfarin. These ones will die. What else do we know? If you're WR, you need a large amount of vitamin K in your diet. If you don't get that, you will actually die from warfarin. So these basically are only resistant to warfarin if they have a load of vitamin K as well. So if you are WR or WS, you will die. So let's go back and find these. So WR, so it's these guys, isn't it, that are likely to die. 56 plus the 8 is 64 of the rats. In a population of wild rats, 51% were resistant to warfarin. Use the Hardy-Weinberg equation to estimate the percentage of rats in this population which would be heterozygous for warfarin resistance. Show you're working. %50 are likely to be resistant. It's co-dominant, isn't it? So we can kind of approach this from either angle. If we say 51% might be here, which leaves us 49% over here. So if Q squared is 0.49, that gives us Q. Therefore we can go, we can work out P. We can multiply 2 times P times Q to get 2PQ. And then we can put that into a percentage form. So 42%. I'm just checking the mark scheme here, see if we can do it the other way around. Yeah, we can do if we need to. That's fine. And then finally, we've got a malaria question. Now there's an interesting thing about this mark scheme I need to flag up to you. Some humans have got a genetic resistance to infection. A recessive allele gives increased resistance to infection from the malarial parasite. In a population, the proportion of babies who are born homozygous for this allele is 0.01. So let's just have a look at our list again. So which one of those are we looking at? It says the proportion of babies who were born homozygous for the allele is 0.01. It's this one, isn't it? Frequency of homozygous recessive individuals, not alleles. So I know Q squared, this is what it's telling me, Q squared is 0.01. Use the Hardy-Weinberg equation to calculate the expected proportion of heterozygotes in this population. So P squared plus 2PQ, we always write this down because you get a mark for it. I know Q squared is 0.01, so I can work out what Q is, the root of 0.01. Therefore I can work out P, therefore I can do 2PQ, and I end up with 0.18. Now, it says the proportion of heterozygous in this population. A lot of you have turned that into 18%, which to be honest I would have done as well. But interestingly, the mark scheme just says 0.18, not 18%. It is an old spec question, and well, it's an old spec question. So I just don't know if we're missing something with that spec. All the other answers are happy with the percentage. So if it says proportion, I don't know what we do, whether we err on the side of caution and do a decimal or both, I suppose would be good. So we could say the proportion 2PQ is 0.18 or 18%, and at least you've got both of them down.